So today we're going to talk about heredity and the basics of genes and genetics. Heredity is the study of how your parents pass down your genes to your offspring, to their offspring. The genes are like genetic code, like DNA. Alleles are specific things that code for your traits. So the different types of alleles that you have a dominant allele and you have a recessive allele. So alleles would be, say it would be brown eyes over blue eyes. So it would be brown eyes. So that would be brown eyes and blue eyes. There's two different types of alleles. There's dominant and then there's recessive. So brown eyes are dominant over blue eyes. So it would be symboled as like a big B to show it's dominant, and then recessive would be a little b to show it's recessive. Dominant, if it's paired up with a recessive, then the dominant trait will show over the recessive. If the recessive shows up and there's no dominant, then the recessive will show. Those are called genotypes and phenotypes. Homozygous means that you have two of the same trait. So you could be homozygous recessive, which means you would have two little b's, or you could be homozygous dominant, which means you'll have two big B's. And if someone is heterozygous for a trait, so we're doing brown eyes and blue eyes, they're heterozygous, which would mean they'll have both, dominant and recessive. But the dominant one would show, but there would still be a carrier of the recessive trait. But the dominant one would show over, and the dominant one showing over the recessive one is the phenotype. So there's genotype and phenotype. Genotype would be the specific alleles and the genetic makeup, which would be big B, little b, and the phenotype is what actually shows, which would be brown eyes. Sorry for my handwriting. Let me rewrite that. Which would be brown eyes. So just to recap, heredity, that's like the sum up of all your genes and stuff that's passed down to you. Your genes is like your DNA makeup. The alleles are the specific traits, like the representation of the traits that you may have, like brown eyes, blue eyes, short, tall, and plants and stuff like that. The dominant would be big B, which will always show over recessive. And someone can have recessive and it not show up because they have a dominant trait but still carry the recessive. But the recessive only shows up, it'll only show up if someone is homozygous. Is homozygous for the trait. That's the only way someone can show a certain phenotype is if they're homozygous recessive. But they can still carry it if they're heterozygous, where they'll be dominant and recessive, but the dominant will always show. So genotype examples would be big B, little b, or if someone has big B if they're homozygous dominant, little b if they're homozygous recessive, and then their phenotype for this one is brown eyes and that one is brown eyes, but if they have this one, it'll be blue eyes. Not blue X's, blue eyes.
Gregor Mendel was an Austrian monk and scientist who tested his hypothesis that traits were passed down from generation to generation through genes. Characters are the categories such as pea plants with their height, like how they're tall, short, white, purple, and the traits are the individual, such as this one's purple, this one's white, or this one's tall, this one's short. And he did this with his pea plants, and he started out with a pea generation of 2 different plants one white, which we're going to represent with a W, and one purple, which we're going to represent with a lowercase w. Now, when these were crossed in the F1 generation, they figured they would blend and it would be a pink flower. Well, to their surprise, they were all white. But when it came down to the F2 generation, the recessive gene, which we now know is the white flower, was passed down because it basically piggybacked on the trait, which they had no clue about. So when the F2 generation, when they were crossed, the F1 generation would have looked something like this, with a purple as the dominant and white as the recessive. The purple showed because it was the dominant. The white did not show because of that. It had to be two, not just one in it. But when it came down to the F2 generation, you had some like this, too small, so the white showed again. All right, law of
Particulate.
law of particulates. Traits that are inherited and discrete units. Law of independent assortment. It is traits separated from one another during gamete formation. And then the law of segregation. It's basically when you're made, you have two copies of the trait inside, and one copy from the mother and one from the father, giving you the two copies. We're going to do some Punnett squares here. We're going to do a heterozygous at the top of the pea plant. On this one, we're going to be doing purple and white flower color. So we're going to put a big P to demonstrate the dominant purple, and we'll put a little p to demonstrate the white. And then on this side, we're going to also do a heterozygous. So we cross these two, dominant, dominant. Cross these two, it's going to be dominant with a recessive, so that is a heterozygous. And then we're going to cross these two and get the same thing. And then down here, we're going to get two small, showing a white flower color, which is a recessive. Now, the genotype of this is going to be 1 to 2 to 1, because we have three different genes here. We have a full dominant homo, we have a hetero, and then we have a homo recessive. So that shows the 1, the 2, and the 1. Now, the phenotype is what they physically look like. So that would be a purple, so that's 1. That would also be a purple because of the dominant, so with that one. So this would be a 3 to 1 because you only have one recessive showing up in the Punnett square. Now down here, we're going to do, say, height for the pea plant. And when I represent tall, I'm going to use the big T, and for short, I'm going to do the small t. We'll also do heterozygous cross again. These two's cross, you of course get tall, dominant, and we're going to get tall with a short. Same thing here. And we're going to get the same genotype and phenotype, because when you cross two heterozygous, you're going to get the 3 to 1 ratio, and you're going to get the 1 to 2 to 1 ratio in the genotype. So that'll be 1 to 2 to 1, and this is going to be 3 to 1. Now, if this was a dihybrid cross, we could actually look and see what we could get to it over here. So this is the possibilities we could have. We could have big P, little t, little t. We could have big P, small p, little t, little t. We could have small p, big T, big T, and so on and so forth. You're just matching genes to what could happen. So you could have a tall with a purple flower, or a tall with a short in this case, or a tall hetero with a recessive on the short gene.
All right, so I'm gonna talk about incomplete dominance. What incomplete dominance is it's kind of like when both traits and alleles blend together to come up with a different trait. So the example I'm gonna use today is red flowers and white flowers. So the red flowers would be the dominant one, and we can represent that with R. And white flowers would be the recessive, we can show with little r. So if a flower would be red, it would be expressed with a homozygous dominant. If the flower is white, it would be expressed with homozygous recessive. But if in a Punnett square and they were crossed, so say we have dominant and then we have recessive. So a red plant has a red plant and a white plant come together, they would make typically these would be 4 red plants, but when it's incomplete dominance or partial dominance, all of these flowers would blend red and white together, and all these plants will now create a new phenotype, which would be pink plants. So essentially what it is instead of one allele or trait being dominant over the other, they share, and so they would now blend together. So now we'll talk about codominance, and codominance is a little different, and this is what you would see kind of like in blood types. So if someone has a blood type of say A, which is dominant, but also BB would be dominant. So instead, like in this case, when these came together, they made pink. These won't come together and come up with a new blood type. They would share. So if we had A, B, B, and a Punnett square, they would all come together and make AB, 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 and they would share. So both A and B would both be equally shown in their blood type. But also, there's still a recessive with blood type, which would be type O. And type O, if it's together in AO or BO, it doesn't show up. It only shows up when it's straight OO. So it would be essentially the same thing. It's just you can only have type O if you're fully type O. But the A and B would share, and they would come together and be one blood type. It wouldn't be just straight A, just straight B. None of them are dominant together, they would share. So different forms of dominance would also be epistasis, polygenetic inheritance, quantitative and qualitative characteristics. So epistasis is kind of like, it's a very conditional type. So an example would be like lab coat or Labradors, their coats. So it would be various differences. So like the combination of, so say H would be for fur. H is dominant for yellow labs, and then say B would be dominant for brown. It would be essentially a dihybrid cross for both yellow and brown that would create a specific form that would give you a black type fur. So you're essentially doing a dihybrid cross for the same trait. And then polygenetic is similar with skin color. So it's kind of like because polygenetic is skin color could be from 9 different combinations, and combination after combination is kind of like the cumulative or collective combination of the genes that you've gotten over time from different ancestors and stuff like that would give you your skin color now. It's not particularly one shade or the next or different types of genes in your specific skin color. It could be a combination of different skin colors that you've developed. And then also another one is quantitative and qualitative. The difference between quantitative characteristics and qualitative characteristics is quantitative are cumulative. So they don't fit in a specific category like skin color. It's like a bunch of different genes that have crossed and crossed and crossed that have created a specific one that doesn't fit in a certain category. And then qualitative would be something that would be measured and put in a group, like tall or short, like purple, white, and plants and stuff.
All right, we're going to look at the mutation sickle cell in blood, and we're going to have two parents that are heterozygous. So we're going to come in with the mother. She's going to be big S, little s, and father, big S, little s. So these two, fine. Comes to here. This is where a carrier is formed. They have the recessive genetic, but the mutation is not shown or apparent. They're just carrying it. Now, when it comes to this one, the recessive gene is shown because there's two of them, and in order to have a recessive mutation, you have to have both genes from the mother and the father.